Hello, everyone. Thank you so much for jumping on with us today. Our webinar will start shortly. We still have some people joining in right now. Please hold tight. We'll probably get started right around another 2 or 3 minutes because we do have some people joining in. Thank you for your patience. And yeah, we'll be hopefully getting started very shortly. All right. Thank you, everyone, for coming in. Just for those of you who are new into the room, just a reminder that we will be getting started shortly because we still have a couple more people logging in and jumping into the room. We appreciate your patience, but yeah, we should be getting started probably in the next minute or so. All right. Well, without further ado, thank you, everyone, for jumping on today and joining us for our webinar, Build Disease Models That Matter, the Power of Custom iPSC Differentiation. Just as a reminder for those who are first timers here, um, we will be holding a Q&A during the end of this webinar, so please ask as many questions as you would like. To do that, we actually have three ways of doing this. So two of the ways, depending upon if you're using StreamYard or LinkedIn to be on here, please submit the questions through the comments section at any point during the webinar. So if you are in, I think the link or in the LinkedIn, you have a comments box that you can click on as well. If you're in the StreamYard, you have a comments button that you can click on to be able to submit through. To help us out, if you can include the slide number for your question, that would allow us to be able to pull back up the slide when we go through the Q&A session. Now, for those who don't want to necessarily have their names out and asking in the questions in the comment sections, you can also send your questions privately through the email questions@elixirgensci.com. Please feel free to send them in. We're more than happy to run through all of them and share them during the Q&A session. But of course, if your question isn't verbally addressed during the Q&A live session, no worries. We'll be able to reach out and answer after the event via email. So please make sure that you're using the name that you're using in the attending is consistent with the name you use to register so that we can send that information over. And without further ado, I'd like to introduce our guest, our speaker here. Um, so Our speaker, Dr. Tetsuya Tanaka, completed his postdoctoral training at the NIH and at the University of Toronto, where he gained expertise in microarray analysis and genetic engineering of pluripotent stem cells. Previously served as an assistant professor at the University of Illinois and research associate professor at the University of Notre Dame, where he led research showing how substrate stiffness affects pluripotent stem cell fate, how Wnt signaling drives tumor-like growth, and how nanopores can precisely electroporate single pluripotent stem cells, and is currently serving as the VP of Stem Cell Biology at Elixirgen Scientific. And without further ado, I'd like to introduce Dr. Tetsuya Tanaka.
Thank you, Henry, for the nice introduction. And hello there. My name is Tetsuya Tanaka. I'd like to explain what our company is and then what we can offer in this webinar today. And then my webinar is consisting about 4 topics today. In the first part of my presentation, I'd like to summarize what is our core technologies. And then on the rest of the presentation, I'd like to explain using 3 different kinds of specialized cells we developed, how we can use those cell types for the accelerating your research. Before I start the presentation, I'd like to have you think about what this 30 trillion represents for. It's actually associated with the current topic. Then, well, it's actually the number of cells in our body. Our body actually has 30 trillion cells. There's actually a slight variation among the different references, but 30 trillion is the sort of a standard number. And then those 30 trillion cells can be grouped into 200 different cell types. And then amazingly, those 200 different cell types can actually arise from one single cell, which is the fertilized egg. And then today's topic is how we can actually produce those 200 different cell types. Now, I'd like to take you to the first cell differentiation event happens in mouse embryonic development. That actually happened on the 3rd day of the differentiation after the fertilization. Now the single cell divided once and it became 2 cells. And then as you see, the differentiation or cell development actually proceed in a stepwise manner. But while cells divide in this way, there are many things going on inside the cells, and then inducing the different type of genes inside the cells, and then consequently, cells actually start to differentiate. Now the embryo produces the inside and then outside polarity, and then now cells embryo is actually expanded, and then now first cell differentiated actually is called the trophoblast. It's actually important to have the implantation successfully done. And then the remaining cells inside the embryo is called still remain pluripotent. It's called inner cell mass. When we isolate the inner cell mass and then put them on the culture dish, we can establish the cell line remain as a pluripotent stem cell. It's embryonic stem cells. And then recapitulating this developmental process in culture dish is a rather difficult task because we need to have that the cells sequentially express the gene induction sequentially. But as we learn that the differentiation is regulated by the important gene, it's called transcription factors. Once we identify and then introduce the key transcription factors, we can actually directly induce the differentiation. So that's the technique we are applying right now. Now I'd like to show you our core technology. So then if we were interested in developing the protocol to differentiate specialized cells using human-induced pluripotent stem cells, first we identify target phenotype. Let me erase the pointer. And then we will identify particularly the marker expression of the specialized cells, and then also morphology and then essential cellular function. Once we identify those, we go back to our database and then identify the transcription factors that located upstream of the markers we are expecting to see in the specialized cells. And then we come up with the several different candidate transcription factors. And then we will make a cocktail, including those candidate transcription factors, using in vitro transcription, and then produce the messenger RNA encoding those transcription factors. Then we will introduce into human iPSCs and then see which cocktail combination actually upregulate the marker expression. Then we narrow down the list of transcription factors. Once we satisfied with the transcription factor cocktail, then next step, we optimize the culture conditions further using some small molecules, growth factors. Then we try to complete the differentiation process, meaning the expression of the marker in almost 100% of the cells within 1 to 2 weeks after the initiation of the differentiation. Then once we optimize the culture conditions, we try to scale up the differentiation scale. So then we can produce the cells in a billion scale. And then we can ship those cells to our customers. Then, so this slide actually summarizes our core technology or the database I mentioned earlier, which we call the human gene correlation matrix. This data set was generated by our founder of the company, Dr. Minoru Ko, when he had laboratory at the Keio University in Japan. What they did is that they isolated 714 different transcription factors encoding in the human genome, and then introduced every single one of those into human embryonic stem cell. Then they isolate each line and then measure the consequence of the forced induction of transcription factors by measuring the changes in overall transcriptomes. Then those data actually located on row-wise or the x-axis in this heat map. And then they compare the similarity or the differences of those transcriptomic changes with the publicly available database for the human tissue transcriptomes. That's located on the y-axis on row-wise. So then if those two data sets actually showing a higher similarity, then in this heat map, it shows red color. Whereas if those two data sets are not correlated, then the heat map showing either white or blue color. So in this way, we can actually identify which transcription factors can induce the specialized cell type. For example, in this left top corner, we have a very high red enriched area. The tissues located in this area is likely like a brain tissues, like hypothalamus, cortical brain tissues. Those are involved in this area. And then on the transcription factor side, if we look at the transcription factors, then those factors are likely inducing those cells in brain tissues, including neurons and the glial cells. So this is how we come up with the list of transcription factors to drive the differentiation of the specialized cells. Then practically, we can actually isolate, we can establish the protocols to differentiate any 200 cell types in our body. So then we call this our technology as a quick tissue technology. And then using advantage of using this technology is that, as I mentioned, we plan the differentiation process within up to 2 weeks. So differentiation is very fast. And then since the differentiation is based on delivery of transcription factors into pluripotent stem cells, it's actually flexible, meaning like we can differentiate almost any kind of iPSC pluripotent stem cell lines using our protocol. So it can be applicable to a wide variety of iPSC lines or pluripotent stem cell lines. And then at the beginning, we aim at what the functionality we are looking for the specialized cells. At the end, we usually get the functional cell types. I'm coming back to this point in later of my talk. And then our production scale is scalable. We can do differentiation even from a 24-well format to T225 flask format. So then we can produce the billion of cells in a week. So that's our business model. This slide actually summarizes the cell types we have successfully produced, establishing the differentiation protocols. Most of the cell types are about the neuron or the glial type, but just because we got many inquiry of producing those neurons or glial type. But we have one mesodermal tissue, like a skeletal muscle differentiation protocol available. And then for the neurons, we have a cortical excitatory neurons, and then inhibitory GABAergic neurons. And then we have also dopaminergic neurons, and then spinal motor neurons. And then its relative cholinergic neurons. I'll talk about these two cell types later in my presentation. And then we have peripheral sensory neurons, and then also two glial cell types, astrocytes and microglia available. And then we are currently finishing up the differentiation protocol to produce the oligodendrocytes. That's the third glial type in our central nervous system. And then the cells will be available in a couple of months. Then if the cell type of your interest is not listed in this slide, we are happy to collaborate with you and then develop the, your cells of your interest. And then currently we are working on developing two other cell types that's requested by our clients. And then we actually obtained the licenses to the few different cell bank. Mostly our iPSCs are obtained from California Institute for Regenerative Medicine, CIRM. That's because that CIRM actually has a wider variety of iPSCs, not necessarily derived from healthy individuals, but also some patient-derived iPSCs. But as to the Parkinson's disease patient-derived iPSCs, as well as the amyotrophic lateral sclerosis patient-derived iPSCs, we work with the different banks. We have obtained the licenses as well. So then customers do not need to worry about licenses. They can use the cells for their research purposes without worrying about the license. Now, switch gear a little bit. I'd like to talk about our major product, excitatory neurons, and then how we can use these neurons for research. So we typically provide neurons as frozen cells And then after thawing and replating those neurons, they actually reach the maturity within 7 days. And then with a few different medium conditions. And then as you see in this phase contrast images, post day 1, post thaw, up to 7 day post thaw, cells actually quickly change the morphology and then exhibit the neuron-like morphology by 7 days after thawing. And then at this point, when we stain the culture with the specific antibodies, most of the cells express the tubulin beta 3, which is the pan-neuronal markers, and almost 100% of the cells express the tubulin beta 3, and then greater than 70% of the cells express the markers for excitatory neurons, V group 1. And then when we when we put them on co-culture and then measure their electrophysiological activity, those excitatory neurons show the robust synchronized network burst within the third week after replating. Then if we don't culture the excitatory neurons with the astrocyte, still those excitatory neurons show synchronized network burst. It's about a delay of 1 week, but so they actually show very strong network burst. So meaning that within 2 to 3 weeks, our neurons show functional synapses and then forming a network among the other neurons. And then using that approach, we have tested the drug-induced response using a non-compound agonist, antagonist of receptors expressed in neurons. For example, glutamate is the agonist of the glutamate receptor, and then actually it excites the neurons. And as we increase the concentration of the glutamate, we see strong accelerated number of network bursts. But at the highest concentration, we saw reduction of the network burst. We interpret this data as cells are super excited, so then induce excitotoxicity. And then another example, when we add the antagonist of the glutamate receptor, like AMPA and kinase receptor agonist, CNQX, as we increase the concentration of CNQX, we saw a reduction of the network burst. So it is actually showing an expected response. Then taking this approach a step further, we actually collaborated with the Johnson & Johnson and then published a paper 2 years ago in a journal called Cells. With this, so the Johnson & Johnson were interested in using this iPSC neuron-based approach to predict the seizure liability of the new compound. And then at that time, the field was actually debating whether 2D culture or the 3D culture, like organized, with which one culture condition would mimic the actual better predictor of the seizure liability. So we did the comparison between 2-dimensional cultures versus spheroid 3-dimensional culture, and then see the we took the this number of non-compound and then investigated whether which culture conditions show expected response. And then based on this study, so for this analysis, we use the calcium oscillation as an indicator of the drug-induced response. And then we took the number of peaks as parameters to investigate the drug response. And then when we looked at compare the 2D versus 3D culture, based on this study, we found that the 2D culture is actually predicting the drug-induced response better than the 3D culture. We interpreted this data because that the in a 3D culture, penetration of the drug might be problematic. So then the 2D culture would be the better, showing the better outcome. So then, now let me talk about the our motor neurons for the use of modeling the ALS disease pathology. So we actually launched the cholinergic neurons. That's actually close relative of the motor neurons by applying the published protocol, which was actually published through our founder's laboratory. But unfortunately, the original protocol of the cholinergic differentiation produced the lower expression level of the HB9, which is the famous marker for the motor neurons. So we decided to expand our lineup and then produce the motor neurons, produce the protocols to differentiate the motor neuron more efficiently. To do so, we looked at the how embryo differentiate, and then the motor neuron is actually produced from the ventral part of the neural tube, and then the positional cue is actually we actually needed to provide a positional cue to the culture of iPSC differentiation culture. So then by looking at this scheme, we provided the ventralization and then caudalization cues to the culture, and then we ended up with a nice neurons with a nice HB9 expression. So we completed the motor neuron differentiation protocol in this manner. Then we applied this differentiation into both healthy donor-derived iPSCs as well as the sporadic ALS patient-derived iPSCs. And then ALS patient actually suffered from the progressive loss of motor neurons, and then they cannot actually move when the disease actually progresses. So it's actually important model to work with the motor neurons. So here I'm showing the typical differentiation process of our iPSCs in this protocol. And then during the first 3 differentiation period, we didn't see much differences between these 2 iPSC lines. But after the cryopreservation and then thawing those cells for the further cultivation, sporadic ALS-derived iPSC motor neurons actually show poor survival relative to the healthy donor-derived motor neurons. And then when we stain those day 7 motor neurons with the antibodies, marker itself is actually comparable between those 2 lines, but the number of cells we counted actually was about half of the cells relative to the healthy donor motor neurons. Then as an indicator of the ALS disease progression, we became interested in the localization of the TDP-43. I'm not going to go in detail about the TDP-43 function in a webinar, but the point is that when the disease progressed, TDP-43 originally localized to the nuclei of the neurons, but they start to mislocalize in the cytoplasm. So that's the first indication of the disease progression. And then we wanted to see, we could see the mislocalization of the TDP-43, particularly in ALS patient-derived motor neurons. Then by looking at the immunostained images, we started to see some indication of the mislocalization at the lower magnification. And then when we looked at the higher magnification, we saw ALS patient-derived motor neurons have some cytoplasmic staining of the TDP-43. And then we observed that the a similar trend by testing two different antibodies, one staining a total TDP-43, and then another staining the phosphorylated form of TDP-43. And then if I remove the tubulin beta-3 staining, actually cytoplasmic localization becomes more pronounced in these motor neurons. Then when I try to quantify these results by using the software comes with the confocal microscope. I counted the TDP-43 staining as a dot and then compare the total TDP-43 versus cytoplasmic staining of the TDP-43. Then I was able to detect the statistically significant mislocalization of the TDP-43 in the cytoplasm, particularly in the ALS patient-derived iPSCs for both total TDP-43 and then phosphorylated form of TDP-43. So then we think that this could be a useful model to screen some drug targeting the ALS disease in the future. Now, at the last part of my talk, I'd like to explain how our microglia can be used for research. So again, the differentiation protocol for the microglia also ends in 7 days. We provide a cryopreserved microglia as a product. And then by day 7, they exhibit the typical amoeba-like morphology, and then also express the unique markers for microglia, IBA1 and then TMEM119. When we check the marker expression by QRT-PCR, and then compare the marker expression before and then after the cryopreservations, we didn't see much reduction of the other markers, so we are quite happy with this product. And then also microglia responds to known cytokines and then release the, responded to the known cytokine based on the RNA-seq results. And then also phagocytotic activity was when their phagocytotic activity is measured, they show expected increase, particularly with the IL-4 treatment. So we are happy with these results. And then when we thought that the microglia could be actually used to identify some drugs. And then for this purpose, we looked at the activation of the microglia as a sort of a readout. We screened the 891 FDA-approved compound. And then by normalizing the data with the Z-score, whatever the compound that's showing a greater than 2, that's actually a hit. And then we found 11 of those hits. And then surprisingly, 10 out of the 11 actually were involved in the same biological processes. So we weren't even bothered by doing the secondary screening, but we focused on one most promising compound and then used in our microglia culture. And then indeed, the addition of this compound actually increased the phagocytotic activity further. And on the phase contrast image, you see that the microglia is actively phagocytosing those Frodo dye conjugated with the zymosan. Then so far we have been able to produce the microglia from Alzheimer's disease patient-derived iPSCs, and it could be useful for modeling the Alzheimer's disease progression. And then finally, we were able to culture 3 different types of cell in one culture conditions. In this image, we have a pink tubulin beta-3 stained neurons, and then GFAP stained astrocyte, and then GFP tagged microglia in the same culture list. And then since the AD progression has something to do with the activity of microglia, establishing this culture system would be useful to study the AD progression. So I'll actually stop here, and then Henry can talk about our business model.
Awesome. Thank you so much, Tetsuya, for the wonderful presentation there. And just a tail end on this talk before going into our Q&A session. As Tetsuya mentioned, we do have more of a partnership solution approach here at Elixirgen Scientific. As you've seen from the great data that Tetsuya shared, we've been able to work on differentiation of multiple different cell types for different groups out there that need it for research. But one of the great tools that we have because of the great uh, human gene expression correlation matrix that we have, if there's ever a cell type that anyone here has been in need of but haven't been able to identify or find, we actually offer a cell development platform where we do partnerships to develop new cell types to be able to develop certain testing or new cell model development altogether, as well as being able to scale up for larger screenings through our cell supply differentiation agreements. We also do assistance with translational research for those that may need to offshoot some of that research type or may not have a lab to be able to do so. We're happy to assist there, especially with some of the great data I know Tetsuya shared in regards to some of the functional analyses that we've done, as well as actually a new avenue that we have grown into, which is iPSC derived cell therapies. And if there are any questions in regards to any of these, please feel free to reach out to us and we'll be happy to set up a call to discuss. Now, with that, I think we can move into our Q&A session. Now, just as a reminder to everyone here, as we shared before, if you have any questions you would like to ask Dr. Tanaka, please place it into the comment section, either into the tab, I think for the StreamYard, and then into the box section in the LinkedIn if you're logged in into either of those. I think we've actually received some questions using the questions at elixirgensci.com. But once again, if anybody wants to send their questions in through there, so it's not necessarily stated in the comments with your name, we're perfectly fine to do so. So I'll let a few of you be able to add in some. Yep, exactly. As in the banner below. Please feel free to send your questions there as well. All right. Okay. So I think we have a couple of questions. Dr. Tanaka, maybe we could start off with this one. So we got a great question where they asked, how does the transcription factor based approach for differentiation compare to other differentiation protocols, including NGN2?
So of course we cannot disclose our neural inducing factors, but as I mentioned earlier, our differentiation protocols using transcription factors, so then we can accelerate the differentiation process. We can skip the precursor stage or what's happening during the earlier differentiation stages. So then we can accelerate the differentiation process. And then usually we observe a strong functionality, as I mentioned, that the strong robust network formation. Our cell type shows the greater functionality, and then it can be applied to many different iPSCs. It's not the protocol to work only limited number of iPSCs, but we have been able to produce the neurons, different cell types using a variety of iPSC lines, including both healthy donor-derived iPSCs and then also disease patient-derived iPSCs. So those are the advantages.
Perfect. No, thank you so much for clarifying that for our attendee. Okay, so I think we just got another question through the email. Have you tested non-sporadic ALS patient lines?
for
I'm guessing in regard with,
non-sporadic. Okay. Yes. So we have actually worked with the 3 different kinds of ALS patient lines with the known mutation. One TDP43 mutation, and then C9orf72 mutation, as well as SOD1 mutation. And then yes, we have actually worked with those 3.
Perfect. So hopefully that answers the question for them there. All right. All right. So from the question, one of the questions we got via email. So it says, you mentioned that your iPSC differentiation method is flexible. Can you please share how many different iPSC lines Elixirgen has successfully differentiated to this date?
So we have done a couple of hundred iPSC lines, including both healthy and disease patient-derived iPSCs.
Okay. Yeah. And as you mentioned before, I know we're always happy to take on any and all human iPSC or human ES lines that anyone would need to be differentiated. I know we're always open to that challenge, right, Tetsuya? Perfect.
Of course.
And then, yeah, so I think, so the last question I have here via the email, once again, if anybody has any additional questions, please add it into the comment section or through the email. But for the last question I see here, what type of drug screenings have you done? In particular, is there a difference between testing small molecules and or synthetic compounds using your system for testing? Interesting.
Could you repeat the first part?
Oh yeah, sure. So the first part is what type of drug screenings have you done or Elixirgen has done? And then the second part is, have you seen a difference between small molecules versus synthetic compounds in your testing?
Okay, so we have the FDA approved the 891 compound library. We bought it from a commercial source. And then I'm not sure whether I'm not sure if I understand the difference between small molecule and then compound.
Well, have we done both by chance, Tetsuya?
I use the small molecule and then compound as interchangeable. So I thought it's the same molecules, but I think we have worked with the both kinds.
Okay, so I think that we can definitely assist. Or we, I think we're pretty open to being able to test out a multitude of different compounds, synthetic or just generic, small molecules and other drugs of options. More of just we just need to have them sent over to test. Does that sound about right?
Yeah.
Perfect. Okay, yeah. So we have another question here. For the J&J study, you co-cultured the induced excitatory neurons with primary astrocytes. Was there a reason you didn't use iPSC or derived astrocytes?
So great question. At that time, we didn't have a protocol to differentiate astrocytes yet. Then it was actually middle of a development phase. So there's no way we could use the iPSC-derived astrocyte for that study. It's unfortunate. We wish to have the iPSC-derived astrocytes for that publication. But now we have a capability to produce the astrocytes using iPSC. So we can actually repeat the same experiment. I didn't have time to mention that actually, but we have actually did the same network burst assay using iPSC-derived neurons co-cultured with iPSC-derived astrocyte.
Yeah, and if anyone would like that information as well, please let us know. We're more than happy to schedule a quick call and can definitely share that information as well as whatever else may be of interest to know. All right. Let me check on the emails. All right, I'm not seeing any more emailed questions. Are there any other questions that anyone in attendance would like to ask? Please feel free to ask in the comments. Um, but if not, um, with that, I think we would like to thank everyone who attended and joined us here. As a reminder, after this, we will be sharing the recording. So if anybody wants to go back and review through all the great and wonderful things that Tetsuya was able to share today. And once again, we'd like to just thank all of you for attending. If you have additional information you'd like to get, please feel free to join or look through our website, as well as feel free to email us either at the questions at elixirgensci.com or the email info@elixirgensci.com. And we'll be happy to follow up in regards to any questions that you may have, as well as happy to discuss if there's any research needs or research interests you'd like to discuss. We're here to help in any way possible. And with that, we thank you so much for joining us and have a great rest of your day.